Good afternoon, everyone. And also, I would like to welcome the audience from Prague, from Charles University. We are also streaming this lecture all the way there. And it's my great honor to welcome and introduce Joan Steitz in Brno here. And I'm sorry, she's such a great figure that I'm quite stressed to do that. So I apologize for reading the introduction. Joan Steitz is a really huge figure, not only in the RNA field, but also in science. She's a great, I would like to say a female scientist, but it would be unfair to say female scientist. She's a great scientist. And her path was not so easy because she began her studies in the 60s, and at that time there were not that many females studying at universities, and there were no females leading the laboratories. After graduating, Joan started her research career in the group of the famous cell biologist Joseph Gall, which actually convinced her to take on the scientific career. And therefore, she then began the graduate studies at Harvard, where she was the only female out of 10 in the class. And she was the first female doing her graduate studies in James Watson's group. And you probably heard how tough James Watson can be. So it was probably not very easy times. And after that, Joan moved to Cambridge, where she was at the LMB. And that was the times where the LMB was at the height of its fame. So among the future Nobel laureates, and it must have been great times. And after that, Joan was appointed a professorship at Yale, and already very early at her career, she became a member of the National Academy of Sciences and was awarded a number of prestigious awards, over 70 if I counted that well. And if I was to go through all these awards, it would be much longer than actually Joan's own talk. So I will just list some of them. So in '83, as I said already, very early in her career, she became the member of the National Academy of Sciences. In 2004, she was awarded the RNA Society Lifetime Achievement Award. She was also awarded the Rosalind Franklin Award for Women in Science. And I would end up with a very recent award. I think that doesn't sound so great, but I think it's very good for a scientist to be awarded a postdoctoral mentoring prize for her long-term mentoring investment into new generations. She's also a member of numerous scientific boards, and I should mention actually what's the most significant about her career is that she, from the beginning, knocked down all the barriers. She was the first one to bring up the concept of RNP particles, and that goes throughout all Joan's career. Then she discovered what's the function of small nuclear RNAs, and then in the future years, she studied a number of other non-coding RNAs, their processing, how they assemble the RNP particles, and I'm sure that we will hear a lot about that today. And I would like to end up that Joan has been the role model for all females in science, but not only for females. And I'm really happy to welcome Joan here. Thank you.
So thank you so much, Stepanka. That was wonderful. Thank you very, very much. Now we can all go home. So this is my first time in Brno. I've been in Prague before, but this is just absolutely amazing to be here in such a historic place. And thank you all for coming. And as you notice, the title I've got here is a little bit different from the one that was advertised, but basically it's sort of a common title that I can use to talk about whatever happens to be going on in the lab at the time. So as you see, non-coding RNAs can be made either by cellular genomes or by some viral genomes. Certainly not all viral genomes make non-coding RNAs. But regardless, the question is the same. What are they doing? And when you think of cellular non-coding RNAs, I think just about anything's possible. Could be controlling anything inside of a cell. For viral RNAs, the possibilities, I think, are a little bit more limited because you would think that a viral non-coding RNA would be doing something to advance the life cycle of the virus or to defend the virus against all the barriers that the host tries to put up against viral infection. Also, since viral genomes are small, if a virus is going to devote part of that precious genetic information to making a non-coding RNA, you would think that it must be doing something fairly important for the virus. And finally, with respect to viral non-coding RNAs, since viruses and their host cells are constantly exchanging genetic information, you can expect to find things on viral genomes that maybe look like something that the host also makes. But it doesn't mean that it's doing the same thing because viruses are very clever. They pick up these pieces from their host cells and then change them in ways to make them better to serve the purposes of the virus. Okay, so what I'm going to do is give you a little bit more introduction about non-coding RNAs, and then I'm going to tell you two current stories from the lab, one about a viral non-coding RNA that we've been working on or puzzling about for a very long time. And secondly, I'm going to tell you about a whole new class of cellular non-coding RNAs that we stumbled into relatively recently. So let me start. I love this slide from John Maddock, the Australian bioinformaticist. What he's done here, and it's almost 10, or it's more than 10 years old now, is to plot the amount of non-coding DNA in the genome of various organisms, which obviously you can't read. So let me tell you what they are. And the title is that the amount of non-coding DNA rises as a function of developmental complexity, which is sort of a mystical term, but I think you all sort of know what that means. So all the blue guys down here are single-celled either eubacteria or archaebacteria, and you notice that they have up to only about 15% non-coding DNA. The black guys here are single-celled eukaryotes, so yeast and fungi of various sorts. The organisms in here are invertebrates and plants, more non-coding DNA. And as you know, when we get to vertebrates and mammals and humans, we have a whopping 98% non-coding DNA. What's an even more profound realization that's occurred really just within the last 10 years or so is that all of that DNA seems to be at some point in the life of the organism transcribed into RNA. So that means that there's a huge number of different kinds of non-coding RNAs made, particularly by these higher organisms. And although the RNAs that are produced can be rather low in abundance or perhaps rather short-lived, that doesn't mean that they can't be playing important roles in signaling or regulation of cellular metabolism, gene expression, et cetera. And of course, that's what we want to try to find out about this vast array of non-coding RNAs. Now, it does turn out, and Stepanka mentioned some of this in her lovely introduction, that in the past several decades, we have learned about the functions of many classes of non-coding RNAs. And these, of course, are the most abundant ones that we were able to study even before the sequencing revolution, where we could look at things that are very, very low in abundance. So let me just go through this quickly to just sort of set the stage. She mentioned the snRNAs, the small nuclear RNAs, that are the parts of the snRNPs with their associated proteins that are the building blocks of the spliceosome that recognize the ends of introns and form this big RNA protein machine that removes introns from pre-messenger RNAs. But they're also non-coding RNAs that are involved in various stages of transcription. In the nucleolus, in our cells, there are several hundred different species of SNO RNAs, small nuclear RNAs. whose function it is to guide to precise locations covalent modifications in the ribosomal RNAs as they're being generated and packaged into ribosomes. In germ cells, we have piRNAs, at least one function of which seems to be to prevent transposons from moving around and wreaking havoc with the genomes in germ cells. And as you probably know, every week, every journal, there's a new paper about some long non-coding RNA that associates with chromatin and brings in usually repressive factors like the polycomb repressive complex that binds to exist, or sometimes positive factors, and influences the expression of whole sets of genes. Once we move out to the cytoplasm, there are, of course, the classical non-coding RNAs, ribosomal RNA and tRNAs involved in translation, and you all know what they do. There's also the experimentalist tool, silencing RNAs that we have used to knock things down, and now CRISPR-Cas is more frequently being used, and a huge class of microRNAs, these tiny little 22 nucleotide long RNAs that associate with specific proteins, particularly agoproteins, sit by partial complementarity on the 3' UTRs of at least 50% of our protein coding genes and regulate either translational efficiency or the half-life of the messenger RNAs, and thereby play very important roles in things like development, differentiation, disease, et cetera, et cetera, et cetera. But as I mentioned, this is only the tip of the iceberg because these are the abundant ones that we knew enough about to have studied in the past. And as I mentioned, there are lots more out there. A couple other comments. Note that a number of these where we know about how they function actually use base pairing between the non-coding RNA and the target in order to identify and carry out their functions. And it also wants saying that virtually all of these are very tightly bound by partner proteins. Stepanka mentioned this in her introduction, and those proteins then are very integral to the function of the non-coding RNA. So they're really non-coding RNPs. Okay, at this point, I'd like to go ahead and, because the first story I'm going to tell you is a viral non-coding RNA story. Maybe this wouldn't be so noisy if I put it in the other pocket. I'm going to introduce you to the viral non-coding RNAs that we've been studying for the last couple of decades. So these are made by gamma herpes viruses, which infect human or other primate cells. Gamma herpes viruses have long double-stranded DNA genomes, enough to encode 100 to 200 genes. They are all lymphotropic viruses, so they infect T cells or B cells. They are all associated with oncogenesis, and they all have both lytic states and latent states. Latent states where the DNA of the virus disappears into the host cell, only a few functions are made, and then something can trigger the virus to go into lytic phase and start making lots more virus. What you notice from the dates over here of the discovery of these non-coding RNAs or RNPs, and I should mention these are the viruses, and over here is how we're doing on function. So if you compare these to the number of question marks that are left over here, you realize that finding functions for non-coding RNAs is not easy. It's very challenging, takes a long time. But recently there's been a real uptick in our ability to define functions. And I think that that's because technology has finally caught up. And nowadays, there are all sorts of wonderful techniques where we can look at the associations of non-coding RNAs with various other proteins, chromatin, et cetera, in cells. And it makes a huge difference to know sort of where to start, who the interactors are in terms of finding functions. So as I said, I'm going to tell you just one story about a viral non-coding RNA, and it's going to be about what's called the EBER2 RNP, which was discovered way back in 1981. The paper that I'll mostly talk about was actually published last year, but it took so long to get there. I really like talking about it, so I hope you'll put up with me on that one. So what we've discovered about this non-coding RNA is that both what it does and how it does it were sort of unexpected. Namely, what it does is interact with a host transcription factor and guide that transcription factor to certain sites on the viral DNA. And it does this in a way that hadn't been described previously for long non-coding RNAs, which is by base pairing with nascent RNA, namely transcripts that are coming from that region of the DNA. And this is the work of Nara Lee, who is a fabulous postdoctoral fellow in the lab. He now has his own lab in the medical school at the University of Pittsburgh. And Walter Moss, another postdoc in the lab, who's an RNA structure, bioinformatics type of person, and Teresiario, a technician. So let me introduce you next to a little bit more about EBV, Epstein-Barr virus. So it turns out that whether or not we know that we had mononucleosis as teenagers, 95% of us carry this virus around in our memory B cells in a latent state. So we're virtually all infected by EBV. And as you probably know, and I mentioned that these viruses are oncogenic, cancers can develop from cells carrying EBV, actually both lymphomas, many types, several different types of lymphomas, and carcinomas. What's shown over here is very important background for this part of the talk. This symbolizes the double-stranded DNA genome of EBV. And what's important here are these things at the end called TRs. So that stands for terminal repeats. So this linear double-stranded DNA genome of EBV has its ends, each end, 5 to 20 copies of a tandemly repeated sequence that's about half a kilobase long, the same sequence. And it's through those sequences that when the virus goes into the cell and establishes latency, you get recombination. It's not terribly well defined, but it clearly involves the terminal repeat sequences to form the circular double-stranded DNA episome that's in the latently infected cells. And then after the lytic cycle is turned on and the virus undergoes replication to make lots more. When the virus is packaged into virions, it's within the terminal repeats that you make the cut to give you back linear double-stranded DNA genomes. So this is just to make the point that these terminal repeats are very important for several aspects of the replication, the DNA replication of the virus. So the RNA I'm going to be telling you about is EBER2, whose picture is over here, EpsteinBarr encoded RNA number 2. And I mentioned when they were discovered, they are RNA polymerase III transcripts, about 170 long, remarkably high abundance in the either latently or lytically infected cells, about 10 to the 6th copies per cell. So you probably know ribosomes are 10 to the 7th copies. SNRPs are 10 to the 6th copies. So this is way, way, way up there, which is why they were discovered in the first place. Actually, EBER1 is a little bit more abundant than EBER2, which is maybe down at 2 times 10 to the 5th or something, but it's still tons of these molecules. They're strictly nuclear. Other labs have shown that they are tumorigenic. If you put them into certain types of B cells, they cause an oncogenic phenotype. And over the years, we've figured out proteins that bind to them. Obviously, geneticists went in and deleted their genes from the genome and didn't see much of a phenotype or would have known years ago what these RNAs are doing. Okay, so the technology that made a difference with respect to figuring out what EBER2 is doing was a methodology called CHART. And that stands for Capture, Hybridization, Analysis of RNA Targets. And it was devised by Matt Simon, who is now an assistant professor in my department at Yale. But he was a postdoc in Bob Kingston's lab at Harvard at the time he worked out this method. And basically what it is, I think you all know what CHIP is. You localize where a protein is sitting on chromatin. This is exactly comparable, except you're localizing where an RNA is sitting on chromatin. So you start out the same way. You do some formaldehyde cross-linking. You shear the chromatin down into little pieces. And then instead of coming in with an antibody, as you would do in CHIP, to select the DNA fragments that have been cross-linked to your protein of interest, you come in with an antisense oligonucleotide that's complementary to the RNA that you're interested in, and that's attached to a bead, and you use that to pull out the bound DNA fragments. You work them up for sequencing, and then do the sequencing, and that way you can tell where the RNA was sitting on the DNA. In order to do this technique, though, there's one absolute prerequisite, and that is that you have to have some portion of your RNA of interest that you know will hybridize to a complementary oligonucleotide that you're going to attach to the bead to use to select it. So the first experiment that Nara then did was to make DNA oligonucleotides, and here are their numbers, that sort of tile along EBER2 RNA. He also tried this with EBER1, but it didn't succeed, so that's why we don't know more about EBER1. But in the case of EBER2, then you can take a cell extract from latently infected cells that have EBER2 RNPs in it, throw in these DNA oligos because there's RNase H in the extract. You will get, and RNase H cleaves the RNA strand of a DNA-RNA hybrid. You'll get cleavage of the RNA at the sites where the DNA that you've thrown in can hybridize. So in this way, Nara found out that there were two regions on EBER2, these two, that were available for interaction. And luckily, he chose this site to attach an oligonucleotide to the beads and do the pullout, because as you'll see, the interesting biology is going to be going on in this other available region over here. Okay, so now he could go ahead and do the chart experiment. And when he did that, I sort of expected that he would see EBER2 sitting various points on the host chromatin. That turned out not to be the case. The host chromatin was virtually background level. But on the viral genome, and here's another control with uninfected cells, and you see that there's very low background, but there were lots of peaks on the viral genome, but a huge peak at the end where those terminal repeats that I told you about are. Okay, so if you blow this up, you would say that EBER2 is sitting on, could be sitting on each terminal repeat. Now I have to admit that we don't really know that EBER2 isn't sitting all on one terminal repeat, and the other terminal repeats don't have any. But this is the way this sort of data is classically handled if you're dealing with repeat sequences. And besides, the numerology comes out wrong. And this got very exciting when Nara discovered that there was a report in the literature of something else sitting on those terminal repeats, giving a very similar profile, and that was a host transcription factor called PAX5. And this is a paper from Paul Lieberman's lab at the University of Pennsylvania. So what is PAX5? PAX5 turns out to be a really, really important transcription factor in B cells. Has a homeobox, looks like a normal transcription factor, but it turns out that it's very important for the commitment to the B cell lineage. It acts at various steps along the way to mature B cells. And interestingly, in both a positive and negative way, that's been documented in a vast literature. It's also essential when you finally get to mature B cells out here. So what the Lieberman lab had done after they discovered PAX5 sitting on the EBV genome was they knocked it down and asked what happens to latent EBV gene expression. And this is another figure from their paper, but the point here is that interestingly, some of the latent genes went up and some went down. So this transcription factor isn't just doing upregulation, it can also be doing downregulation, and that'll come up again later. Okay, so we have this important transcription factor from the host sitting on the terminal repeats apparently at the same place as EBER2 is sitting. Are they actually physically interacting? And we had the tools to do this. We had antibodies against PAX5, so we could ask, do they pull down EBER2? Yes, they do, in immunoprecipitation. And we also had these nice antisense oligos linked to a bead to pull out EBER2, and they pull out PAX5. But I should say right away, we already had indications at an early point that this is not a direct interaction. I won't go into the details there, but that there were other proteins in between PAX5 and EBER2, and I'll mention those at the very, very end of my talk. Okay, so they're structurally interacting. Are they also functionally interacting? And I showed you that the Lieberman lab showed that if you knock down PAX5, one of the partners here, you get upregulation and downregulation of various EBV latent genes. So the next experiment was to knock down EBER2 and ask what happens to the expression of those very same genes. And it turns out the very same ones that went up when you knock down PAX5 go up when you knock down EBER2. So this is evidence for functional interaction. And let me just focus in a little bit more on this region of the EBV genome where this is going on, so you know what the landscape is. It turns out that these terminal repeats in the circular genome end up residing in an intron, which is the first intron of two different isoforms. Here are the downstream exons and the initial exons of LMP2, so it's got A and B forms. And these are genes that are very important for maintaining the latency state of the virus. And there's also another gene that goes in the opposite direction called LMP1, and that's the major oncogene of the virus. So these are very important transcripts during latency. And just to quantitate some of this data a little bit more, if you now look to see how effectively PAX5 gets to the terminal repeats after you've knocked down EBER2, it's significantly down, whereas on a control promoter or a known host promoter that binds PAX5, it's not down. So it's down specifically in this particular context. So the next experiment then was to knock down PAX5 and ask, does EBER2 get to the terminal repeats? And I'm not going to show you the data for that experiment, but I'm going to tell you, that there we were very surprised by the result Nara got, because it turned out that knocking down PAX5 didn't matter. EBER2 could get to the terminal repeats either with or without PAX5. And that was important because it sort of knocked down one of two alternative hypotheses for what might be going on. We already knew, okay, so this terminal repeat sequence is 538 base pairs, and it had within it two PAX5 consensus sequences. So you would think that the most direct way of getting this complex there would be to have PAX5 bind to one of its consensus sequences and bring in EBER2. But I just told you that you can knock down PAX5 and EBER2 still gets there. How does that happen? So Nara came up with another hypothesis, and that was that we know that there is RNA being made from the region. I told you that the terminal repeats are within this large intronic sequence, that maybe EBER2 has a sequence within it where it can use RNA-RNA base pairing to base pair to this nascent transcript. And in that way, EBER2 gets recruited. It brings along with it PAX5, which can then find its consensus sequences and sit down on them. And if that's true, then you predict several things. First of all, you have to have a good RNA-RNA interaction between EBER2 and the terminal repeat sequence. You should be able to block that RNA interaction, and it should give you the same phenotype as completely knocking out EBER2, just covering over that sequence. You should require transcription through the TRs, because the nascent transcript is involved, and you don't have the nascent transcript unless you have transcription going on, and you should be able to get physical evidence for an RNA-RNA interaction. And the next bunch of slides I want to run through is just quickly the evidence that Nara, and in this case Walter Moss, got for these predictions being true. So Walter used a program called RNA Up that looks for extensive sequence complementarities between two RNAs and found that indeed within the terminal repeat sequence was complementarity to EBER2 that maps up here in this region of EBER2. And in fact, one of those sequences sits very close to a PAX5 binding site. But remember, the transcription factor is going to bind to the DNA, whereas we're presuming that EBER2 is interacting with the RNA. So this isn't competitive, this is complementary. And Nara then made a morpholino oligonucleotide that you can put into cells that would just base pair with this region, cover it over, prevent it from interacting, and that gave the same phenotype as the actual knockdown of EBER2 on these latent, the expression of these latent genes. A really wonderful way of inhibiting transcription from a specific locus by Pol2 is a variation of the CRISPR-Cas methodology that was worked out in Jonathan Weissman's lab. And basically what is done here is to make a guide RNA that brings the complex to a particular site on DNA, but you haven't you've inactivated the active site of Cas9, so it doesn't cut the DNA, the complex just sits there. And what they showed was this blocks RNA polymerase II from getting through, so you don't make nascent transcripts that are downstream of that. And so Nara made guide RNAs to the two first exons of LMP2, the A and B exons, and they were very effective at knocking down transcription downstream. And when he then looked at the amount of EBER2 that got to the terminal repeats, that was significantly down as an effect of blocking transcription through the region by this nice trick. If you want to show that they're direct RNA-RNA interactions, the method of choice is to use sorolin, this molecule that intercalates into usually imperfect double-stranded structures in RNA and also DNA, and then forms covalent cross-links between you, but can also work with other residues at a particular 365 nanometer light, but can be reduced at reversed at 254. Nara first set up the situation in an extract, making sure that he could see a cross-link and that it was reversible with 254 nanometer light, and then went in vivo and did the experiment and showed that you preferentially bring down terminal repeat RNA after doing sorolin cross-linking and pulling out with a probe against EBER2. So what I've told you is that we think that this is the right model for how this interaction is occurring because depletion of EBER2 results in decreased PAX5 localization to the TRs. It's EBER2 that's bringing it into the DNA site, that there is potential base pairing with the terminal repeat, which is blocked by an antisense morpholino oligo, sorolin evidence for RNA-RNA interaction, and you need to have transcription through the terminal repeats in order to get this to happen. So, great, this is how the thing acts. But why? What's the reason for having this interaction with the terminal repeats? And here we went back to thinking about the terminal repeats and their importance in DNA replication, both during latent replication and lytic replication. I thought it would be more likely to have something to do with latent replication. Nara checked that and there was no effect. But when he then checked lytic replication of EBV, first by knocking down EBER2, as you see here, and then basically this is an old-fashioned southern blot, just showing the amount of EBV DNA produced in the control knockdown cells and the EBER2 knockdown cells. You see it's definitely less. And if you do something more modern like PCR, you see that the amount of EBV genome is significantly down. But if you look at the amount of EBV DNA that gets into virus particles, that's really quite significantly down as a result of knocking down EBER2. And likewise, you expect knockdown of PAX5 to do exactly the same thing. So we used 2 different shRNAs against PAX5 that effectively knock down PAX5. And here you see again, there's a significant diminution of the total amount of EBV DNA replication. But if you look at the viral titer, it's much more significantly down. And the reason that we find this exciting, and we will be working on this in the future, is that the clinicians tell us that before the onset of an EBV-induced tumor, there is invariably a round of lytic replication of the virus. So this may be the link between the oncogenic phenotype of EBER2 and DNA replication. And obviously we have to figure out what the steps are there and what's really going on. So let me just finish this part by saying that, if this is really true, there are a lot of EBV-like viruses out there, you would expect the situation to be the same in another one of these. In fact, we know of lots and lots and lots of viruses that infect other types of primates that are closely related to EBV. There's a phylogeny. The problem is that sequencers don't like to mess with repeat sequences. So we only have repeat sequences, as it turns out, for 3 of these, the ones boxed in red, those sequences at the ends. It turns out that the marmoset virus has lost its EBER2, does it in some other way. However, this rhesus monkey lymphocryptovirus has an EBER2 and has terminal repeats, so we can look at that situation. And here's its EBER2, can fold into the same secondary structure, but it's only 65% identical sequence-wise. In this virus, the terminal repeat is 933 base pairs long, completely unrelated sequence, but there is one PAX5 consensus sequence binding site, and where the putative interaction maps, again, is very close to that. So the same model would apply. And we think this is, pretty encouraging evidence that what I've been telling you probably is what's going on. So let me conclude by talking just a little bit about what this is saying about possible interactions between long non-coding RNAs and their targets and what they're doing. So there are 2 models for long non-coding RNA action that have been pretty much pushed in the literature, and there are several cases that are examples of each. One is as a scaffolding function that proteins bind, and those proteins need to act together in the function. And an example of that would be the telomerase RNA or the 7-SK-RNP, where you need to have those proteins brought together. And another example is the example of EXIST, where you have it interacting with the chromatin and bringing in a protein or protein complex that then regulates the gene activity of a region of chromosomes. But this model, where a non-coding RNA interacts with the nascent transcript by RNA-RNA base pairing, and in that way brings something into the chromatin, is a sort of new and different model for how a long non-coding RNA might act. And we sort of thought, well, gee, there must be other instances. So I was really excited about a month ago to talk to Scott Bailey, he's a structural biologist at Johns Hopkins. And he's been doing structures on a CRISPR-Cas that's not the one that we know so much about, that's from a different bacterium. And in his system, it turns out that transcription through the guide region is essential for the cutting by the CRISPR system. You have to have ongoing transcription. And what he's figured out is that it looks as if the guide RNA is pairing with the nascent transcript. That brings in Cas9, and then Cas9 is activated by this interaction in order to cut. So this might be a completely comparable type of situation. I suspect there will be more. So let me finish this part, and I'll guarantee you the next part isn't as long. by talking a little bit about the open questions that what I've just told you raises. Some of them are sort of mundane, and some of them are pretty profound, I think. Why would a transcription factor need a non-coding RNP to guide it to its consensus sequence in DNA? Why can't it just get there? Well, we really don't know the answer to this question. It could be that it's secluded in some way, or it needs it for stability. I don't know. This is one of the things that Nara is going to work on in his new lab, because he comes from a chromatin background. Another sort of curious question, although there are precedents, is why does reducing the level of a transcription factor up the expression of neighboring genes? And here I want to mention the work of Thomas Jennewein, who's in Freiburg, in Germany, who's been studying transcription factors, many that belong to the PAX family, that bind to repetitive elements in our DNA, like signs and lines and so on. And what he's found is if a transcription factor seems to bind sort of independently and not with a whole bunch of other transcription factors the way you have at a promoter, but if there seems to be just a single transcription factor binding, that seems to be repressive on the chromatin. And it might be because we have these repetitive elements here that we're looking at something that's sort of the same, however that works. That might help us. I mentioned we have no idea how you get from binding to the terminal repeats to the regulation of lytic replication, but that's something that we're definitely going to be looking into because it's important for tumorigenesis. Since Nara left for Pittsburgh, he's done a great job of identifying actually 3 proteins that are the intermediaries between PAX5 and EBER2 interactions. They turn out to be RNA binding proteins that are also components of perispeckles. But the problem is that perispeckles are these nuclear bodies. They have a long non-coding RNA in them called NEAT and lots of different proteins, but nobody knows really what they do. So that doesn't help us that much. But where it does help us is that in thinking about where did the virus get this idea of using a non-coding RNA to bind to nascent transcripts and bring something into the chromatin, because there might be cellular non-coding RNAs that act just like EBER2. And now that we know what some of these other proteins are that bind directly to EBER2, we can do IPs with those and look for cellular non-coding RNAs that might be the cellular analogs of EBER2, and we'll see what happens. Okay, so now I'm going to go on and talk about a completely different project, which is the discovery of a whole new class of human non-coding RNAs, although they seem to at least be in mouse, and I suspect they will be in other organisms, where transcription is induced downstream of human genes. In this case, what's been most thoroughly studied is biosmotic stress. And these transcripts, which were discovered by Anna Wilberg, a Swedish postdoc in the lab, fabulous, she calls them dogs for downstream of genes. And she was helped by a wonderful undergraduate who's now in an MD PhD program at Rockefeller, by Kaja Tukowski, who's a longtime research scientist in the lab, and again by Therese Yario. So when Anna came to the lab, obviously she didn't set out to discover a new class of non-coding RNAs. She had come from the Karolinska, which you probably know studies cancer, and she'd worked with neural cells. And she had read in the literature about a couple of long non-coding RNAs that were associated with bad prognosis in neuroblastoma. And she wanted to select one of these and study it. They'd just been said that they existed. Nobody knew what they did, and she wanted to study them and figure out what they did. And so she started out by treating neural cells with KCl, which she knew depolarized the membrane and turned on transcription, and then looking at the expression of these various loci that had been described as giving rise to long non-coding RNAs in the literature. And one of them, this one called 145, was incredibly induced, 40-fold here, 45-fold here after 4 hours by the treatment with KCl. She then very rapidly discovered that it wasn't the membrane depolarization. It didn't have anything specifically to do with neural cells because it happens in other cells, and that what she was really looking at was an osmotic stress response due to adding KCl. And so here I show you other osmotic stressors like sodium chloride or sucrose also give this very, very large induction of this particular transcript. So it turned out, okay, so let me just mention at this point that these osmotic stress conditions are not extreme. Some people in the literature use up to 1 molar when they do experiments on osmotic stress. So this is only 80 millimolar. And we think that this is sort of more physiological, perhaps mimicking the type of stress that an organism might experience if they have kidney failure or severe dehydration or something that's, you know, within the physiological range. Let's see, what was I going to say next? Sorry. Oh, yes. Okay. So it turned out that this locus that had been described in this original paper wasn't part of any annotated transcript. So in order to figure out what was going on, Anna decided that she should just do deep sequencing of the transcriptome, both before and after KCl, and then look at the region in which this RNA 145 mapped. And it turned out that it resides downstream of an annotated protein coding gene called CXXC4. And what you see is that on the strand that has the normal transcript, not on the other strand, after osmotic stress, you see an incredible increase in reads, both in the total reads and in the spliced junctions, the map spliced junctions, downstream of this gene. And this is a huge region. This is about 60 kilobases. So in this whole region, you're seeing RNA transcripts where you weren't seeing them before. They're only at about the 10% level relative to the upstream annotated transcript, but they're definitely different from what you see if you haven't done the osmotic stress treatment. So she had, you know, not only data on this gene, but global data, and the question is this happening downstream of any other genes in the genome, or is this one gene in particular? And of course, it turns out that it's happening downstream of other genes, not all genes. So this is the bioinformatic sort of pipeline that she followed. Please don't ask me about it. But what she ended up concluding was that in 3 different biological replicates, where she only counted the ones where there was 95% coverage over the first 10 kb, she could come up with about 300 of these extended transcripts going downstream of genes, whereas if the criteria were a little less stringent and she just asked for 95% coverage over the first 5 kb, then there were a couple thousand of these dog downstream of gene transcripts, and that means about 10% of human protein coding genes, since we have about 23,000 of these, have the potential to give these transcripts downstream of the locus after osmotic stress. It turns out that in fact you do see some reads in these regions without in untreated cells, but they definitely go up both in terms of mapped reads and in terms of spliced junction after KCl. So we're talking about a lot of what are called have been called intergenic transcripts in the sort of, you know, RNA parlance. So Anna had some evidence that these were, oh, okay. So the question was, you have this upstream annotated gene, and you have this downstream region where you're clearly seeing many more transcripts. And so what's happening in these 2 different regions? So she chose 6 different genes that gave rise to these dogs and quantitated by PCR how much the downstream region was upregulated, and you see values ranging from 10-fold to 90-fold. as opposed to the upstream region where the most you see is twofold for one of these genes. So it looks like this is a read-through phenomenon, but I'll get back to that in just a moment. And in order to tell you what sort of molecular level we're talking about here, the undergraduate Maria did a reconstruction experiment and concluded that before KCL for Anna's favorite dog, this CXXC4 one, there was about 1 copy of this downstream transcript per cell and 30 after. So where are these transcripts in cells? Unfractionated cells into cytoplasm, nucleus, and then the chromatin fraction from the nucleus. And what you can see here for different ones of these dogs is that they seem to be in the chromatin fraction as compared to controls, whereas the upstream, the annotated polyadenylated RNA that gets out to the cytoplasm actually does end up in the cytoplasm most abundantly most of the time. But remember, since only about 10% of these are actually continuing on, what I've just told you is not inconsistent with each other. Okay, so where are they if they're in the chromatin fraction? And obviously what we wanted to do was some sort of fish experiment, fluorescence in situ hybridization experiment, and we decided to invest in Stellaris fish probes, which some of you may know are very expensive, but they tile along your gene of interest and are very effective in allowing you to do localization of a relatively low abundance RNA. And what Anna saw, this is now another dog producing gene, she would see up to 4 spots per nucleus after osmotic stress. So that's consistent with having a diploid genome, which she knew these cells had either before or after DNA replication. And in order to ask where these 4 dots are, we also got Stellaris probes for 2 of the introns of this particular gene. And the overlay then very strongly suggests that these transcripts are staying very close to the site of transcription because you're seeing the merge color there. Okay, so the next important question, and particularly referees really liked this question, was, are you initiating new transcription downstream of the gene in this big 60 kb region, or are you really elongating through? And Anna did a bunch of experiments. I won't show you most of them. This is, I think, the most convincing one, where she used a technology that had actually been developed by another postdoc in the lab, Mingyi Tsi, where we used EIF4E protein. So that's the translation initiation factor that binds caps, 5' caps. So if you take a population of RNA and fragment it, and then pull out just what binds to EIF4E, you get all the capped fragments. And you can then map them. And what you see here is that the cap fragments both before and after KCL map to the 5' end of the upstream protein coding gene. There aren't a whole bunch of new ones down in the region where the dogs are coming from. So as I said, this is the strongest piece of evidence. What we're looking at here is some sort of failure of polyadenylation termination that allows these transcripts to continue on downstream. And we don't know anything more really about the mechanism, but obviously that's got to be figured out. One possibility was that you're actually making these dogs at all times, and they're just getting turned over, whereas the upstream part of the gene gets cleaved, polyadenylated, sent out to the cytoplasm. So to look at that, Anna added actinomycin D, looked at a couple of dogs, and saw that the half-life of those regions was no different before and after KCL. And she also did a sort of more modern experiment by adding ethinyluridine, a pulse, and then you can use click chemistry to link that to biotin and pull out just newly transcribed RNA. And you see that the new transcription is huge in the downstream regions and virtually not at all in the upstream gene. Finally, and this will be of interest to some people here, if the polymerase has to get through this huge long region, you might expect that it would be less abundantly populated by cleavage and polyadenylation sites that lead to termination. And indeed, when you look at the frequency of AAU, AAA, and the second most common poly-A site, they're less abundant in these very stringent dogs than they are in control genes that just don't give rise to dogs. They have more of those signals. We did try knocking down various cleavage and polyadenylation factors and got a small increase in dogs, like maybe twofold, threefold, fourfold. So it's not the whole effect, but there's something there probably that's going on. So, okay, so now we sort of come to the nitty-gritty question, which is, you know, are cells just doing this because they're under stress, because you've thrown all of this high salt at them? Or is this something that they're doing intentionally and that's going to help them get through the stress? And here, Anna reasoned that she knew that KCl releases intracellular calcium, and she got her undergraduate, Maria, to then look into calcium chelators and what they would do with an extracellular calcium chelator, EGTA. There's no big effect on the production of dogs. But if you use this membrane permeable calcium chelator that gets into cells called BAPTA, you definitely do not get the dogs. So you have to have intracellular calcium released in order to do this. And what's known there is that it's released from the ER after KCl treatment. And one thing that's involved in regulating that is the IP3 receptor. And farther downstream then in the same pathway are calmodulin kinase and PKC and PKD. So the next experiment was then to add an inhibitor to the IP3 receptor and ask what would happen. And what you see in this slide is that inhibiting the IP3 receptor, you also don't get dogs. So the IP3 receptor signaling pathway is definitely involved. Maria also did experiments with inhibitors of PKC and PKD. Those weren't quite so clean as this, but it turns out that if you inhibited them both those and calmodulin kinase, that then you had a big effect. So we think that this is under the control of one of the known cell signaling pathways, and therefore it looks like a physiologically important response. So just a little bit more. So what do dogs do? Why are we making this RNA? What you've seen is that they're highly inducible after osmotic stress. They're localized to the chromatin, and at least a large fraction of them seems to reside at the site of transcription. It turns out that they arise from all over euchromatin. So down here is plotted on the 23 or 24, or however you want to count them, human chromosomes, where all the ref-seq genes are and where all the dogs are. And you notice they're both just all over, except there don't seem to be any dogs made by genes on the Y. But then there are only about a dozen protein coding genes on the Y. All the rest of it's repetitive elements and stuff. So maybe that's meaningful. Maybe it's just, random numbers. What we know from the literature is that osmotic stress causes nuclear shrinkage and funny things happening to the chromatin. And there's a couple of papers, but one very nice one from a couple of years ago from Jeannie Lawrence's lab, implicated the large sort of bulk of non-coding RNAs that arise from repetitive elements in the genome, like the signs, ALUs, et cetera, in stabilizing the nuclear scaffold, maintaining euchromatin. And so the idea that we came up with was that maybe this bulk of RNA is contributing in the same sort of way to preserving the chromatin structure during this period of stress. And the very last data slide that I want to show you is here. When you go in the literature and ask, okay, how do people assess nuclear integrity, chromatin, et cetera, it's not terribly, what should I say, biochemical. What you do is you look at your cells with DAPI staining. So you're looking at the DNA. And so what you're seeing here is a field of cells that have been untreated compared to a field of cells that have been treated with KCl. So dog production is coming on, and they don't look too much different. Or if you add inhibitors to the IP3 receptor. So I should say in Jeannie Lawrence's experiments, the way they knocked down this massive transcription was either by RNAs, adding RNAs or adding transcription inhibitors to cells. But we had a much more specific tool because we could block the IP3 receptor and not get the dogs being made. So that's what you're seeing here. And if you do that, and then you get undergraduates to sit down and look at these nuclei and plot how many holes they have and what the area is. The nuclear area definitely is smaller. There's shrinkage if you don't have dogs because you've inhibited them. And if you try to map the, how many big holes and nasty looking things are going on in the nucleus, also you see an effect there. So obviously, this has to be done with something much more definitive, but we think that's the direction it's going in. So in summary then, what I've told you is that these dogs are chromatin-associated transcripts. They're generated by read-through after stress. If you think about it, these things can be turned on much more rapidly if you just stop terminating than if you had to start new transcripts and assemble things at a promoter, which would take time in order to make these long transcripts. I've shown you that they're regulated by the IP3 receptor, and these other pathways are implicated. There are many, many of them. Many of them are absolutely huge. So they do explain a significant fraction of intergenic transcription. And of course, the question is, can we really show that they're doing something positive with respect to the nucleus and chromatin after stress? And what I should say is since our paper came out, Anna has been collaborating with several people to look at heat shock, another kind of stress. Do you make dogs? Yes, you make dogs. Oxidative stress is another undergraduate who's been working on that. Yes, you make dogs. But the analyses are just now being done to ask, do you make exactly the same dogs? We don't think so, but largely overlapping genes give rise to the dogs. And I should say that if you try to look at which genes make dogs by GO-term analysis or something, there's no rationale to it. So it looks like other types of stress also give you this phenomenon of this unexpected transcription downstream of genes. And even in virus-infected cells, there's a nice paper from Lars Dolken in Germany who's been studying herpes simplex virus-infected cells, sees transcription downstream of genes, downstream of host genes after herpes simplex infection. So there might be viral stress, there might be cancer stress, who knows? There's just lots and lots and lots of things to look at. So I just want to end by showing you this slide. So when Molecular Cell was about to publish our paper, they said, do you want to submit something for the cover? So Gilbert Garbisco, undergrad in the lab, drew this lovely potential cover. So here's the end of the gene. The dogs are breaking through the barrier, continuing on downstream. Here's the polymerase making lots more RNA. And we thought this was a shoo-in for the cover. Molecular Cell didn't like it. They published something with insipid green blobs on the cover that month, but I like showing it nonetheless. Okay, and here are the people who've been responsible for the bulk of what I've told you about. This is Nara Lee, who is now at Pittsburgh, and Walter Moss, Anna Vilberry, Maria Passarelli, Taggio, and I am listing here all the various people who've been in the lab, who've worked on the EBERs over the years. There have been lots of them because there's been 3 decades involved. And it's just awfully nice to finally get to the point where at least you know something about what one of them does. And we definitely are working on the other one in our funding. And thank you very much. And I hope there's still a few moments for some questions. Thank you.
So That was really amazing. Thank you very much. And I think this only confirms what I just said in the introduction, that if someone was coming to hear about your old research about the snRNAs, it just confirms that you have such interesting data coming out of your lab all the time. And I'd like to also give you an honor. It's a MENDEL medal for all your achievements, and also for coming to give the lecture.
Well, it's a real privilege to be talking in this fabulous room.
So thank you very much.
Thank you. Oh, wow. Oh. It's very lovely.
And I'm sure there will be many questions.
Thank you for a very nice lecture. My question, well,
yes.
I have maybe comment to one of your open questions. If I can see maybe a parallel of your case in telomerase, which is in fact recruited by transcripts, telomeric transcripts, interaction with the telomerase RNA, and proteins bind to it, like including catalytic subunit and all the other proteins. So it may be similar.
I think you know more. Okay, so Tara, I mean, I knew that there were these transcripts that go, but they actually recruit telomerase?
Well, it is one of their potential functions. This is quite unclear.
Okay.
What is their actual function?
Okay,
it's
but
rather inhibition.
that's another
or
possibility.
activation.
Great, thank you.
And a related question, what's your opinion on the origin of telomerase in eukaryotic cells?
Okay, the origin of telomerase. And you mean, was there some viral origin, or? I'm of the school that thinks that cells probably had to come, or some sort of rudimentary cell probably had to come before viruses, because viruses are so dependent on their cells for making proteins. You've got to have ribosomes, you've got to make proteins, and you can't do that without cells. So I guess I would think that, you know, the evolution of the various reverse transcriptases probably went pretty much hand in hand, and the viruses ended up with some of them and the cells ended up with some of them. I mean, you know, we could talk about this all night and we'd never get anywhere, because, you know, you can't answer these questions about evolution. Do you have a different opinion? No. Okay. Mary.
I was just wondering, what proteins bind to the dogs? Are they similar proteins? Have you looked at this?
We haven't looked at that yet, but that's a great question. Yeah. I mean, the other thing we want to figure out is, and if anybody has any good advice for me, if you could isolate the polymerases that are sitting on the dog regions as opposed to the polymerases that are upstream, would they have different associated factors? Anna's done a little bit with looking at a couple of chromatin modifications and didn't see any differences. And it is clear that there's, of course, more Pol2 sitting on the dog regions after osmotic stress than before. That's clear. But in terms of the details, there's got to be something about those details. Yeah.
John, I was wondering, you mentioned a couple of the stresses, and of course I would be interested whether DNA damage might be some of the stresses you looked at.
Okay, well, we haven't looked at that many. And that is something that we looked at oxidative stress, peroxide. But DNA damage in general, we haven't looked at, but it definitely should be looked at. What do you think?
That's what I would expect.
You think it might be like all the other stresses?
Yeah, in particular, maybe replication stress inducers.
Yeah.
And with that, maybe a similar question. Did you try to look at the cell cycle changes? I mean, if that would be something.
No, we haven't. But other people must have looked at cell cycle changes after particularly heat shock. I just don't know.
No, meaning, you know, whether there is a change in the role of these factors.
Of dogs at different points in the cell cycle. Yeah, so I mean, the half-life of these guys after osmotic stress is about like 2 hours. So they go away after you remove the osmotic stress within about 2 hours. It turns out that for the heat shock ones and the oxidative stress ones seem to hang around longer. So they're clearly slightly different, and the kinetics of their onset is also slightly slower with these other conditions. So there are clearly different things going on there, and that could have to do with cell cycle, but who knows?
Actually, you said that these dogs are depleted of poly-A's. Does it?
Oh, I didn't. Okay, they're depleted. The regions that make them are depleted in poly-A signals. I didn't actually say. If you just do poly-A selection on them, they're about 50% of them seem to have poly-A. And I don't know whether the other 50% don't because they don't or because they're still nascent transcripts, so they haven't gotten them yet. That we don't know.
Okay, and do you see the different dogs between different treatments, like heat shock proteins? And
well, that's what we're currently analyzing. We finally have the data from the same cell type, having done osmotic stress and heat stress and oxidative stress, and just now the data are being compared to see to what extent they overlap. So I don't know yet, but we'll know very soon. But as I said, I think there'll be a large amount of overlap, but not complete overlap, because we already know about some genes that get turned on by one condition and not by another.
Actually, one more question. Do you know what
Sure.
EBER1 does? Because it has very similar phenotype.
Yes, I know. I would love to know what EBER1 does. I want to know what EBER1 does before I die. We've worked on it much too long to not know. I don't know. I don't know. It binds a whole bunch of proteins, and maybe it's just sequestering some important proteins and changing cell metabolism, and that's what it does. I think that's pretty dull. It'd be much more interesting if it interacted with something, you know, really interesting and significant.
So when I will have also a question. When you say that after removal of the osmotic stress that the dogs disappear, do they relocalize, or are they degraded on the chromatin?
You're the one who's going to find out how they're degraded. I don't know. We don't know anything about their decay.
And did you try to look whether the osmotic stress has any effect on the 3' end processing machinery? Whether you said
or
that
the 3' end processing machinery. No, okay. So because we thought it was involved, we've tried knocking down various components of it and don't see the huge effect that you see if you do osmotic stress. But no, we haven't looked to see whether osmotic stress affects 3' end processing. But haven't people done that for heat shock? I'm pretty sure somebody did that for heat shock a long time ago, looking at the effect. And maybe there was some effect on, could it be on histones? Okay, but histones don't have dogs. We've looked at that because people keep asking about that. So histone, there aren't dogs downstream of histone genes. And I think histones processing doesn't work very well after heat shock. Oh, we know that, yeah. Histone processing does not work.
They have a different type of processing, slightly different type. They don't have the classical
yeah, right, they may, right. I don't know. I'd have to go look in the literature. But you can discover what's degrading dogs. That would be wonderful.
Okay. More questions? So if not,
everybody's
I would like
tired.
I would really like to thank you very much again for coming and
thank you all for being here.
There is an apero, I believe, after so please, students, you can still go. Not only students, go and talk to John. And thank you very much for coming.